Our next speaker is Joe Heitman, who's a James B. Duke Professor and Chair of the Department of Molecular Genetics and Microbiology at Duke. He's the Director of Tri-Institutional Molecular Mycology and Pathogenesis Training Program. His research focuses on microbial pathogenesis, antifungal drug targets, and the evolution of sex and sex determination in fungi, and the roles of unisexual reproduction in microbial pathogens. He's been a Burroughs Wellcome Scholar. He received Angem, Squibb, and the NIH Merit Awards. He's been an HHMI investigator, is a fellow in the ASCI, AAM, AAAS, the American Association of Physicians, and is the editor of multiple journals. Dr. Heitman received his degrees, his undergraduate degrees at the University of Chicago, and his MD and PhD degrees at Cornell and Rochester, respectively. And he's been a fellow at the Biocenter in Basel, Switzerland, where he discovered TOR as a target of rapamycin in collaboration with Mike Hall and Rao Mava. I'd like to ask Joe to come forward and talk to us about where he sees mycology today. Thank you, Joe, for joining us. You'll see your existing slide and the coming slide. Okay, we've got a pointer there if you need that.
Well, I want to begin by thanking Pete Pappas and the other organizers for the invitation to come to Asilomar and participate in the meeting. And I think we also owe a debt of gratitude to John Perfect for the analogy for the evening session to a Christmas carol of the ghost of Christmas past, the ghost of Christmas present, and the ghost of Christmas future. So I want to cover 3 topics today with you about mycology today and frame how a basic scientist thinks about the field and what we need as the infrastructure to advance clinical applications. And so the first topic we're going to cover is a revolution in genomics of human fungal pathogens over the last 10 or 15 years. The second topic is species recognition and naming. And then finally, some recent genetics advances. So some 15 years ago, Jerry Fink convened a very small meeting at what was then the Whitehead Institute and became the Broad Institute. And it was called the Fungal Genome Initiative. And the premise was to level the playing field for people working on any pathogenic fungi, whether it was a pathogen of humans or of plants, by sequencing the genomes to the highest resolution possible, including the annotation of all the genes within the genome. And it began with just that very simple premise that we should make this resource available to the community. And so many sequencing centers and many investigators have participated. And so this is the fruits of labor of hundreds of individuals. So this is one of the very first papers in this series on the genome of Aspergillus fumigatus. And it was compared with a sibling species that's a non-pathogen, revealing whole sets of genes that are missing or diverged in the non-pathogen. And many of those have gone on to be implicated in virulence of Fumigatus. And it was one of the very first applications of microarray approaches to look at transcriptional programs as a fungus adapts from the environment to the host, to look at the impact of temperature on the organism. Now, another landmark paper in this series was work led by Geraldine Butler from the community and Christina Cuomo at the Broad Institute, sequencing the genomes of 8 Candida species. And this includes Candida albicans and Dubliensis and many others. And you can see that some of these are parsed out as diploid organisms and others as haploid organisms. In some of the diploid organisms, there's large tracts of loss of heterozygosity as the genome is adapted to this diploid state. And one of the general findings was there have been dramatic changes to the mating type locus with implications for which of these species have extant sexual or parasexual cycles that give rise to genome exchange. Another general feature to looking at the genomes of these organisms, they've all undergone an event where the genetic code was changed from CTG encoding leucine to serine. And the vast majority of those CTG codons were lost in this pathogenic group, and then many new ones arose. So the whole genome was essentially rewritten in response to changing this one codon of the genetic code in the CTG clade of Candida species. Now, we're going to continue, and this is the first paper on the genomes of Coccidioides and some of the closest relatives of Coccidioides. This is work that Jason Steitz was critically involved with in John Taylor's lab. And one of the general conclusions was that many of the enzymes involved in degrading plant carbohydrates in neighboring species have been lost. You see they're all gone in Coccidioides immitis and Posidacea. But there's been retention or expansion of enzymes involved in utilizing peptides or amino acids as a carbon source. And so essentially there's been a shift from organisms that live on plant material in the environment to growing on carcasses. And it was thought that that was a prelude to these organisms evolving to be able to infect live animals. And so you can see the pathogenic adaptations that have occurred across the whole genome as these saprobes of the environment have evolved to be pathogens of animals. This is a comparative genomic analysis of Paracoccidioides, and there were 3 isolates that were sequenced here, 2 from Paracoccidioides brasiliensis and 1 from Paracoccidioides lutzii. And there was also an optical map that was generated for one of these to provide a very high resolution view of the genome. And again, you see that in Paracoccidioides, as in Coccidioides, there's been a shift from utilizing plant-derived carbon sources to proteins. And they took a closely aligned sibling species that's not a pathogen called Uncinocarpus ricei and conducted actual growth experiments using a biolog apparatus where they could look at growth on dozens of different carbon sources and showed that there was a shift from using plant-derived carbon sources to utilization of amino acids and peptides as carbon sources. So again, a shift from fungi that live on plant material to fungi that live on animal-derived material. In addition, there's an expansion of a kinase family that's completely unique in fungi called the FunK1 kinase family. We can go on then to look at the genome of Blastomyces and the closely aligned species of Emantia. Again, you'll see this is work led by Christina Cuomo at the Broad Institute in collaboration with Bruce Klein and many others from the community. And they sequenced 3 isolates of Blastomyces dermatitidis, an isolate of a new sibling species, Gilcrestii, and then 2 Emantia species, Crescens and Parva. And one of the things you can see is that the Blastomyces genome has been dramatically expanded compared to the neighboring species. They all have genomes of about 30 megabases, but in Blastomyces it's been expanded to up to 80 megabases in the genome. And there are large regions of low GC complexity throughout the genome that are just filled with transposable elements. So the Blastomyces genome has just been expanding and expanding and expanding. And what's not clear is why the transposon repertoire has become so much more dramatic in this species. Is there a transposon that's gone wild, or is there control mechanisms that have become disabled, leading to the expansion of the genome? They see that there are a large number of lyse M domain proteins that are involved in chitin binding in the genome, and again an expansion of this kinase family, the FunK1 protein kinase family. And in addition to sequencing the genome, they also did broad transcriptional analysis looking at expression in both macrophages and from mouse lung. And there are several general conclusions that you can see. One is that a large number of proteins involved as antioxidants are induced, factors involved in metal ion acquisition, and then again this theme of switching to using amino acids as carbon sources, particularly the cysteine catabolic pathway. Now I want to tell you a bit more about the Cryptococcus pathogenic species complex as a prelude to talking some about the species names as well. So for many years we've recognized Cryptococcus neoformans as having 3 varieties, originally Grubii, neoformans, and Gottii. And a couple of years ago, the variety Gottii was elevated to species status as Cryptococcus gottii. So we now recognize 2 species in the complex, and yet we know that there are 4 different serotypes and then a variety of different molecular types that we can see at the whole genome level, including these 4 that are recognized within Gottii. So the Genome Project for Cryptococcus has begun to tackle this complexity. And the very first genome paper published in 2005 was a collaboration between Tiger and the Stanford Genome Center, led by Brendan Loftus at Tiger at the time. And two strains were sequenced, JC21, produced by Jeff Edman, and B3501A, a strain from Jun Kwon Chung at the NIH. And it turns out that these are siblings of each other from a genetic cross. So half of their genome is identical and half their genome is different, just like siblings from any kind of genetic cross. And this turned out to be a tremendous genetic and genomic resource. So you can see here the 14 chromosomes of the genome. Each one of them has a unique region that turns out to be the centromere of the chromosome, where there are fewer genes and fewer expressed sequences, and they're filled with transposable elements. And then the regions of the genome that are identical are in dark blue, and the regions with polymorphisms are shown in light blue. And so essentially creates a molecular and a genetic map for natural variation in the population. So this has been a very rich resource for follow-up to identify virulence attributes. Now the Genome Project's also focused on the sibling species, Cryptococcus gaudii. This is work from the Vancouver Genome Center, led by Cletus D'Souza and Jim Kronstadt, in collaboration with Christina Cuomo and James Galligan at the Broad Institute. And they sequenced two of those lineages within gaudii, a VG1 isolate from Australia from Veal and Meyer, and a VG2 isolate from the Cryptococcus gaudii outbreak that's ongoing on Vancouver Island, R265. And at the whole genome level, one of the stark comparisons was that the VG2 lineage has lost all of its components that are involved in the RNAi suppression pathway. And this turns out to be a rich resource to identify additional components of the RNAi pathway and study the implications of loss of RNAi on genome evolution. Now, Christina Cuomo has gone on now to sequence all four of the major lineages within gaudii, so VG1 and VG2, which were previously reported, now also VG3 and VG4 isolates. So all four of the cryptic species within the complex have been covered at the whole genome level. And then the most recent paper is on the serotype A lineage. So this is now the type strain for serotype A Cryptococcus, the H99 isolate from John Perfect at Duke. And this is again work led by the Broad Institute in collaboration with Fred Dietrich at Duke and Guillaume Jambon at the Pasteur Institute. And this is a series of H99 isolates that have been obtained from labs all over the world. So all of these isolates were sequenced as part of this genome project. And in addition, at both the Broad and the Pasteur, deep strand-specific RNA-seq analysis was done to look at the transcriptional repertoire of the genome under a variety of different growth conditions. And there are two general conclusions then. One is that there's very, very complex RNA biology that's going on in this organism. It's a very intron-rich genome. Essentially every gene has introns within it. There's a lot of patterns of alternative splicing, and there are myriad non-coding RNAs that we're just beginning to explore what the functions of these non-coding RNAs might be for the biology and the pathogenesis of the organism. The other important general finding was that there's been a loss of virulence and also of mating potential with laboratory passage. Some of these isolates are less pathogenic and infertile in sexual crosses. And we could identify the genes within the genome that have changed in response to lab selective pressure that are responsible for that. So I want to turn then from this revolution in the genomics of human fungal pathogens that's covered essentially every species, and now in many cases hundreds of isolates within a species, to talk about species recognition and naming. And the genomes of these organisms helps with this. But a lot of this also just involves classic mycology and also population genetics. So there's been a lot of discussion about changing naming in mycology from the one fungus, one name edict. And so we're no longer going to have anamorphs and teleomorph designations. We'll just have one name for each fungus. And at some level, that's simplifying. At another level, it's complicating because we have to decide which of those names we'll choose. And in some cases, we'll need to change names. Now, one example of this is Penicillium marneffii has now become Tayloromyces marneffii. And you'd think that a change as dramatic as that might not meet with widespread approval, and yet in the case of penicillium, the community has really embraced this, and now I think everyone is comfortable with this name. In the case of Aspergillus fumigatus, this Aspergillus genus is polyphyletic, and so in taking one fungus, one name to its logical extreme, it's been suggested that Aspergillus fumigatus should be renamed Neosartoria fumigata to fit it within that particular genus. And I think that's going to be much more difficult to implement for practical reasons. And this will probably be a source of discussion in terms of people who would like to discuss nomenclature changes. Now, another way to think about name changes is that in many cases where we thought there was one species, it turns out there are multiple species. And I think we're much more comfortable with that. And then the question becomes, I think, how important are these naming distinctions? So one that's now more than 20 years old was the recognition of Candida dubliniensis as a sibling species to Candida albicans. And I think this has also met with widespread acceptance, and we recognize this as a distinct species. In the case of Coccidioides, work from John Taylor's lab divided it into two species, Posidacea and Imidus. And again, these are distinct species based on multiple different criteria for defining fungal species. And it's a useful distinction to make. And that's been welcomed, I think, by the community. And at the whole genome level, you can see, in fact, that these are different species that are rarely, if ever, exchanging genetic information. In the case of Candida parapsilosis, for many years, there were 3 different groups that were recognized. And from molecular phylogenetic studies, we could parse out, and this is work from Frank Odds, including also Neil Gow, and define these now as 3 species, Candida parapsilosis, metapsilosis, and orthopsilosis. And you can see a nice tree here distinguishing these from each other, compared to albicans, dubliniensis, and tropicalis. And there's even a 4th species in this group now, that's Lotoromyces longosporus, which is probably the 4th species in the parapsilosis complex. In the case of Aspergillus fumigatus, there's a sibling species that was recognized by Arun Balaji and Kiran Mar, Aspergillus lentulus, based on both morphological and molecular criteria. And so again, this is now a well-accepted, well-validated species that's more than 10 years old. There are other groups of fungi, though, in which it's going to take a bit more time probably for acceptance. So in Blastomyces dermatitidis, very recently, there's a sibling species that's been recognized called Gilcrestii. In murine model systems, some Gilcrestii isolates are more pathogenic than dermatitidis isolates. There's very low-level genetic diversity in the isolates of Gilcrestii that have been studied so far, and it may reflect a bottleneck or a selective sweep of a more virulent isolate. And I think there's much more work that will need to be done on this species recognition and distinction within the Blastomyces complex. It's similar within Paracoccidioides. There's now 2 recognized species, Braziliensis and Lutzii, that you can see nicely distinguished from each other in molecular phylogenetic studies. But within Paracoccidioides braziliensis, there's 4 cryptic species, designated S1, phylogenetic species 2, PS2, PS3, and PS4. And these haven't been named yet, so there are probably 5 species within this pathogenic species complex. And in the case of Sporothrix, the original species was Schenkii, and this has now been split into Globosa and Braziliensis and Lurii. And in particular, Sporothrix braziliensis is now undergoing direct cat-to-cat transmission and cat-to-human transmission during outbreaks in Brazil. So an organism that we think of as being a saprobic environment associated with plants and plant debris has a sibling species that's undergoing direct animal-to-animal and animal-to-human transmission. And there's a lot of potential impact for the evolution of virulence in settings like that. And these isolates are more virulent in murine models compared to some of the other sibling species. Now, one of the common questions in renaming these sibling species is, are there distinctions that you can parse out clinically? Is it important to recognize them as distinct species? This is work published in PLOS Neglected Tropical Diseases recently from the Brazilian groups, showing that there are distinct patterns of infection caused by Braziliensis compared to Schenkii, and that also their responses to treatment differ. And so not only is it important phylogenetically and scientifically to name them as different species, but there might also be prognostic and treatment implications as well. Now I want to return then to the Cryptococcus pathogenic species complex. And we recognize two species here now, Neoformans and Gaddii, and this wasn't always the case. These used to be three varieties, Grubii, Neoformans, and variety Gaddii. And at one of the Cryptococcus meetings that was held in Adelaide, Australia, there was a very vocal discussion about elevating Cryptococcus neoformans variety Gaddii to species status as Cryptococcus gaddii. And according to fungal nomenclature rules, you're not allowed to elevate a varietal status to a species status. And yet, everyone in the community was calling this Gaddii. And so the only logical conclusion was to break the nomenclature rules and name the species Cryptococcus gaddii. And so Jun Quan Chung and Toon Bukout and others teamed up and wrote a paper to conserve this name compared to other historic names, and this variety became Cryptococcus gaddii. So a species that had three varieties was then elevated to two species and two varieties. So it turns out that's an oversimplified view of the situation. And so Toon Bukout's recently published a paper in Fungal Genetics and Biology proposing 7 species names for this complex. So serotype A strains are globally distributed, most common causes of human infection. And in this nomenclature scheme, they remain Cryptococcus neoformans. The serotype D strains are defined as a sibling species called Cryptococcus D neoformans. And the epithet D means derived from or out of, but it's also a convenient way to remember that these are serotype D strains, D neoformans. The problem then is Gaddii. It becomes Deuterogaddii, so like deuterium, deutero means two. The VG3 group has a historic name, Bacillosporus. The VG4 group is Cryptococcus tetragaddii. And then there's a 5th group that was an AFLP type that in his scheme becomes Decagaddii. So this is a lot of names to absorb at once, and whether or not there are clinical implications for this or not isn't immediately clear. But from a phylogenetic perspective, there's absolutely no question that these are distinct species, and if you were just simply a basic mycologist, you would give them names. So I want to close then by just telling you about a few very recent genetic advances to give you a flavor for how people are applying all of this genome information and recognition of species. This is work from Heitman Advances and Suzanne Noble's lab at UCSF, in which they've sought to capitalize on the whole genome analysis of Cryptococcus. And they've generated from the approximately 6,500 genes in the genome mutations that have been signature tagged with a molecular barcode for just over 12,000 mutants in this cell paper. And that tag allows you to mix them together and do mixed infections and deduce virulence attributes from a pool of mutants. They've expanded this set now to 2,112 mutants, and all of these are available via the Fungal Genetics Stock Center for anybody who wants to work on these. And their explicit NIH-funded goal is to delete every gene in the genome, at least for the genes that are not essential. Now in parallel, for both Cryptococcus and many other fungi, including Candida albicans, other investigators, and this is work from Yong-Sun Ban at Yonsei University in Seoul, are deleting defined sets of genes. And so in this work, he's deleted all the transcription factors from the Cryptococcus genome and then phenotype these under a very broad array of conditions with the explicit purpose of generating networks of understanding how the transcriptome of the cell is wired. And so combining both Hitton's unbiased deletion approach and these more defined deletion approaches, eventually they're going to converge, and we'll have something similar to the Saccharomyces gene deletion collection, in which you can just go into your freezer and pull out a mutation for any gene that you might be interested in. Now, in parallel, the approaches for generating mutations in the genome have been advancing. And I'm going to close with just a few slides that are about the application of the CRISPR-Cas9 system to Candida albicans into Cryptococcus. This is work from Jerry Fink's lab, in which they've engineered the Cas9 nuclease and guide RNAs and been able to go in and engineer the genome to have mutations in the AD2 gene, which leads to these red pigmented colonies, mutate the uracil biosynthetic gene, leading to an auxotrophic phenotype, or introducing mutations in RAS that change the colony morphologies. And this is a very facile system that can generate homozygous mutations in this diploid very rapidly and easily. They've even been able to generate combined homozygous deletions in multiple genes by introducing more than one guide RNA at a time. And most of these systems are set up now in which you can kick out the Cas9 system. So this is work from Aaron Mitchell's lab, in which they expanded the work from Jerry Fink's lab, implementing it in a way in which they can express the CRISPR-Cas9 system transiently rather than in an integrated fashion. So you can delete genes in the genome and then remove this so that there's no heterologous effect on the organism. This is a proof of principle showing they could knock out the AD2 gene. And Aaron went one step further to show that there are two ways in which the CRISPR system is working. In one, it's generating breaks in both copies of the AD2 gene that are replaced by the introduced DNA, so two independent gene replacement events. And in the other pathway, there's an initial gene replacement event, a break made in the other copy, and then gene conversion between the chromosomes to lead to mutation of the second copy of the gene. But the end result is the same, that you generate a homozygous mutation of the target gene. And then very recently, this same sort of approach has been applied to Cryptococcus neoformans, and with one further step that they have a suicide construct in which they can use a guide RNA to delete the CRISPR-Cas9 system following gene deletions at a target locus. And in the case of Cryptococcus, it's a haploid, so you only have to get a single event from these engineering approaches. So I want to close then with the three topics that we've covered to give you a sense of mycology today. That for the genomes of human fungal pathogens, these are essentially available now for all species, and for many of these species for many isolates, and by many I mean approaching hundreds to 500 isolates. So we have just a very robust genomic resource platform. For species recognition and naming, this is a challenge, but I think it's scientifically necessary, and it may have clinical implications. And I think it's important that we recognize these as species, that we name them as species, and then we're going to study them and see whether or not this is going to have prognostic or diagnostic or treatment implications. And then finally, the genetics and recent advances are really enabling a realization of the potential for the genomic resources to have a profound impact here. And the overarching idea is that the more you know about the pathogen, the more we're going to be able to advance the field and think about ways that we wouldn't have been able to envision without that information as the backstop. So I'm going to close with one of my favorite slides here with two of my favorite colleagues in the field. And I'm going to echo this theme that Jack Bennett mentioned, that mycology is a field where it's both collegial and collaborative, and that our strength is really not in the individual members of the community, it's in what we can accomplish working together. And I've been privileged to teach many years now at the molecular mycology course at Woods Hole, and I think that's really emblematic of the theme and the focus of that course that was founded by Jack Edwards and Aaron Mitchell and Pete McGee, and I think that we owe them a debt of gratitude for helping train many of the young people that I see in the audience here today. So I'll stop there. Thank you very much.